like, subscribe and hit the bell icon for prior notifications.
Hello everyone, today I am going to discuss one of the most important events of reproduction, that is fertilization or syngamy. Fertilization is basically a physiological process in which there is fusion of a haploid male gamete with the haploid female gamete to produce a diploid zygote. So basically fertilization is the process of fusion of gametes for the production of zygote, and it is one of the key features of sexually reproducing organisms which produce gametes. And in humans, like other diploid organisms, gametes are haploid. So this is the male gamete sperm and this is the female gamete ovum. After fusion during fertilization, the zygote thus formed contain 46 number of chromosomes. So these are haploid carrying 23 chromosomes, and at the end of fertilization, after fusion of one sperm and an ovum, the zygote contains 46 number of chromosomes, which is specific for human species. That means fertilization ensures the restoration of chromosome number of a particular species. And in humans, fertilization is internal and anisogamous. Internal and anisogamous. Internal means it occurs inside the body of the female, and anisogamous means different type of gametes are fused. Aniso means different kind of. So here you can see the ovum, which is the female gamete, is spherical and non-motile, whereas the sperm is elongated. The male gamete or sperm is elongated, it is microscopic, highly motile. There are so many differentiating features between the male gamete and female gamete in case of humans. So this type of fusion of gametes, the gametes which are morphologically dissimilar, when they fuse during fertilization for the production of zygote, that kind of fertilization is called anisogamous. So in humans, the fertilization is internal and anisogamous. Next, where it occurs inside the human body? It occurs inside the ampullary region or ampulla of fallopian tube or oviduct. Fallopian tube or oviduct. So here I have already drawn a diagram. This is of internal genitalia. This is vagina, vaginal chamber, which is otherwise known as copulatory chamber, where the semen is deposited during copulation or sexual intercourse. And this is the uterus. The lower end is the cervix. This is the cervical canal. This is the main uterine cavity. This is the fundal part, and this is the oviduct or fallopian tube. So this part is the uterine end of fallopian tube. Next, the narrowest part, isthmus. So this is the narrowest part, which is isthmus. Next to it, the widest part, which is known as ampulla. So inside this ampulla part of the oviduct, fertilization occurs. After that, this funnel shaped area is infundibulum, which ends by forming finger like projections which are called fimbriae, and this is the ovary. So from ovary, secondary oocyte will be released during the process of ovulation, and it reaches the fallopian tube, the ampulla of the fallopian tube, by ciliary action or ciliary movement of the internal wall of the oviduct. So next we'll move to how fertilization occurs. We'll begin with the events of reproduction I have discussed earlier. Let me remind you events of reproduction. So reproduction begins from gametogenesis. Gametogenesis is the process of production of gametes. So in case of humans, spermatogenesis is the male gametogenesis process and oogenesis is the female gametogenesis process, as a result of which we get sperms and ovum. So after gametogenesis, there is insemination. after gametogenesis there is insemination. What is insemination? Insemination is the deposition of semen or seminal fluid inside the female genital tract, specifically inside the vagina. So why there is need for insemination in humans? Because the female gamete or ovum or secondary oocyte which is released from the ovary during ovulation, suppose here let me draw, suppose this is the secondary oocyte, and after ovulation it reaches the ampulla. So this is non-motile. Sperms have to come and reach the secondary oocyte for fertilization, and that's why the male gametes or sperms in humans are motile, because they have to reach the secondary oocyte for fertilization. So for that insemination is needed. So during sexual intercourse, about 3.5 ml of semen is ejaculated inside the vagina, deeply inside the vagina near the cervix. So what is ejaculation? Ejaculation is the exit of semen out of the penis, and when it is deposited inside the vagina, then that is called as insemination. So in each ejaculate means one time ejaculation contain 3.5 ml of semen, around 3.5 ml of semen, which contains about 300 millions of sperms, different glandular secretions, in which the main component is fructose and different enzymes. So the semen collected inside the vagina at the end of sexual intercourse contain 300 millions of sperms about, and glandular secretions. The main component of this secretion is fructose and different enzymes. The fructose here present in the glandular secretions provide nourishment or energy to the sperms to remain alive inside the female reproductive tract. So this is the main energy source for sperms. So what happened next inside the vagina? We know that the vaginal environment is highly acidic, and in that highly acidic medium many sperms are destroyed, and only about 200 sperms they find their way through the uterine cavity and they reach the surface of the oocyte. So what happens inside the vagina after insemination and before fertilization? We'll only discuss here pre-fertilization events, and in another session we are going to discuss the main events of fertilization. In this session we'll discuss only up to pre-fertilization events. So after insemination, what will happen inside the vaginal chamber? So inside the vaginal chamber there is sperm destruction. So what are the reasons of this destruction? Number 1, acidic medium of vagina, because sperms can survive only in the alkaline medium. The glandular secretions, which are the composite fluid secreted from the seminal vesicle, prostate gland and Cowper's gland, they try to neutralize this medium. So while they are neutralizing, many sperms are destroyed. So that is the main reason. Next is due to the presence of phagocytes or leukocytes in the vaginal wall. Next, abnormal sperms are also destroyed. There are so many abnormal cases of sperms, double-headed sperms, genetically abnormal sperms. They are also destroyed. And only about 200 sperms, they find their passage or way through the uterine cavity to reach the surface of the secondary oocyte. And during sexual intercourse, the sperms from the vaginal chamber, they are taken up into the uterine cavity by continuous contraction of the smooth muscle layer of the uterus. So there is continuous contraction of the uterine smooth muscle layer during sexual intercourse, and the sperms here, they are taken up into the uterine cavity, and the sperms from here, from the uterine cavity, they move through the fallopian tube and they finally reach the surface of the oocyte. So here the secondary oocyte has been released during ovulation. Remember, after ovulation, within 24 hours, fertilization should occur, because we know that lifespan of secondary oocyte after ovulation inside the ampulla is only up to 24 hours. That means only within that 24 hours fertilization should occur. If fertilization does not occur within that 24 hours, then possibility of fertilization during that month ends. So there is no possibility of fertilization further. That means conclusion is that all insemination during a month do not lead to fertilization or do not end with fertilization, and that insemination should occur as early as possible after ovulation. So now what will happen? There is capacitation of sperms. Remember, there is capacitation of sperms, which is one of the main pre-fertilization event. So what will happen now inside the uterine cavity and fallopian tube? So there is capacitation of sperms. So this is the physiological maturation of sperms. That means the sperms which are deposited here in the seminal fluid, which enter the uterine cavity, they have no ability to fertilize the oocyte. They gain their ability to fertilize the oocyte inside the female reproductive tract only. So that process of gaining ability to fertilize the oocyte is called capacitation. So what is capacitation? It is the physiological maturation, or by which the sperms are made capable to fertilize the oocyte. So that process is called capacitation, which is usually done by the female reproductive tract secretion. So during this process, what happens? There is destruction of the coating substances present over the sperm head, especially over the acrosome. Remember the structure of the sperm. So over the head, the Golgi body is modified to form a cap-like structure, which is called acrosome. So over the acrosome, the plasma membrane of the sperm has many receptor sites. So when sperms move through the uterine cavity and then fallopian tube, with the help of the secretion from female genital tract, those coating substances are removed from the head, and the receptor site present over the sperm head or over the plasma membrane of acrosome, so that those are exposed for different physiological activities of fertilization further. So what happens during this process? There is exposure of receptor sites present over sperm head for further action, and this is achieved by the action of the secretion of female genital tract. So let us summarize the topic. So fertilization or syngamy is the process of fusion of haploid male gamete with the haploid female gamete to produce a diploid zygote. In case of humans, the male gamete contains haploid number of chromosomes, that is 23, and ovum also contains 23 number of chromosomes, and at the end the zygote contains 46 number of chromosomes, which is specific for human species. And in humans, fertilization is internal and anisogamous, and it occurs inside the ampulla of the fallopian tube. And we are here only discussing about the pre-fertilization events up to capacitation, means the sperms, the capacitated sperms have reached the surface of secondary oocyte. Up to that, the main events of fertilization we are going to discuss in another session. So first we have studied the events of reproduction. It begins with gametogenesis process. So we have sperms and ovum at the end of gametogenesis. Then there is insemination. Insemination is the process of deposition of semen inside the vagina during sexual intercourse. The semen, about 3.5 ml of semen after the end of sexual intercourse, is ejaculated or deposited deep inside the vagina near the cervix, and it contains around 300 millions of sperms, different glandular secretions, in which the main component is fructose, which provide nourishment to the sperms to survive inside the female reproductive tract, and also it contains different type of enzymes. But there is destruction of sperms inside the vagina. Many sperms are destroyed, only about 200 sperms survive, and they find their way to reach the secondary oocyte for fertilization. So what happens inside the vaginal chamber? The sperms are destroyed due to high acidic medium or environment of the vagina, and then the presence of phagocytes or leukocytes inside the vaginal wall, inner wall of the vagina, and abnormal sperms are also destroyed. So after the end of this process, only 200 sperms left, about 200 sperms left, and they are capacitated inside the uterine cavity and inside the fallopian tube by the secretion of the uterine cavity wall and the fallopian tube wall. So what happens during this process of capacitation? That means these only 200 sperms out of these 300 million sperms, they gain their ability to fertilize the ovum. So that process of gaining ability is called capacitation. Otherwise you can say that this is the physiological maturation of sperms. Now the capacitated sperms reach the surface of secondary oocyte near the corona radiata layer for fertilization, and the whole process of capacitation takes about 5 to 6 hours. Remember, all inseminations do not lead to fertilization. So as the lifespan of secondary oocyte is of 24 hours only after ovulation, fertilization should occur within that 24 hours. So as early as possible after ovulation, insemination should occur and sperms should reach the surface of secondary oocyte. So now the capacitated sperms have reached the surface, and now the main events of fertilization begins. So that is the acrosome reaction, which is the beginning of fertilization process. The capacitation leads to acrosome reaction. So we are going to discuss the acrosome reaction and the further steps of fertilization or events of fertilization in another session.